warahmatullahi wabarakatuh. Welcome back to my channel. I hope you know well. Myself, Nabiha Nadeem. So in this particular video, this is actually the part 2 of the topic which we were discussing in the previous video, and that is the cell, the structural and the functional unit of all living beings. So in this video, we'll be discussing the second content of the cell, or the second part of the cell, and that is the plasma membrane. We've already discussed the cell wall in the previous video. This video will give you an idea about the plasma membrane or the cell membrane. Okay, so in this picture, as you can see, this is a complex kind of structure of a cell membrane. Let's start from the very basics. Forget this picture now. Let's start from the very basics. Focus on this picture abhi right now currently. Okay, so let's start with the basics. Like what plasma membrane or cell membrane is. So every cell is bound by a thin delicate membrane which is called the cell membrane or the plasma membrane. It is the outermost covering of an animal cell while the second outer covering of a plant cell. Means it is common between both animal and plant cells. It is common between them, but it is the outermost covering of an animal cell, but the second outermost covering means this one of the plant cell. And I already told you the reason in the previous video. If you have not watched the video, you can watch it on my channel. Okay, so it is a covering of the plant cell or the animal cell which separates the content of a cell from its outer environment. Okay, so the plasma membrane being flexible helps it in engulfing food and other substances from its external environment, and this is what endocytosis is. Okay, right, right to understand it in a better way, consider plasma membrane to be the balloon. Okay, a balloon filled with water. Consider this balloon to be filled with entire water. Now just take all the features which this balloon has and compare it, or relate it with the plasma membrane. It is thin, it is flexible. Yeah, it is thin, it is flexible, it is containing the water or the fluid kind of material. We can relate it with the cytoplasm which is present in the cell. Okay, like if we prick the balloon with a needle, the entire content will come out of the cell, it will burst out, kind of. So the similar case is with the plasma membrane also. It is flexible as like the balloon it is, and it is flexible in nature, it is thin, it is containing the entire content of the cell. Okay, now, apart from these features, plasma membrane is selectively permeable. Therefore, it allows only selective substances to pass in and out of the cell. It is selectively permeable. Like in the cell wall we were talking, so cell wall was freely permeable, allowing all the substances to pass in and out of the cell. But talking about the plasma membrane, because plasma membrane is common in both animal and plant cell, so it has to maintain its rigidity, it has to maintain its permeability, so that selective permeability, so that any invader or any foreign substance might not harm the cell, isn't it? Yeah. The second point is it bounds the semi-fluid content of the cell. That means the cytoplasm content of the cell, it bounds that, as I already told you. Third point is it protects the cell from injury and provides an outer boundary to the cell. Clear? Okay. Fourth point is it allows the flow of material and information between different organelles within the cell, as well as between one cell or the other, means the adjacent cells. Very clear? Yup. Okay. Now talking about the plasma membrane on a generalized basis, like how we came to know about plasma membrane, the existence of plasma membrane and all. So there was a model in 1972. In 1972, this model was given by Singer and Nicholson. So they gave a model which was called the fluid mosaic model, and this mosaic model, they also gave a statement. The statement that was given by them was proteins are like icebergs in the sea of lipids. Proteins are like icebergs in the sea of lipids. Okay, now let's see how. Okay, see, now focus on this picture please. this is the simplest picture that I could have shown you of a plasma membrane. Now see, this kind of chain is present in the plasma membrane. Okay, now here you have the phospholipid. These are what? The phospholipids. We have the phosphate head and the fatty acid tails. And the feature of this is that this is polar and hydrophilic, and non-polar, that is hydrophobic. You know what is the meaning of hydrophilic and hydrophobic? Let me tell you. Like in the first picture which I showed you of a plasma membrane, in that you notice that they were arranged in this way. Like here also you can notice that they are arranged in such kind of structure. And these heads of the phospholipids, these phospholipids heads are facing the outer environment, and these are also facing the outer environment, but they are inner to that, because they are hydrophobic, means phobia of water, but these are hydrophilic, that is loving water. They love water. Okay, so this is what they are, and that is why they are arranged in upright positions, so that the hydrophilic area can be towards the water, and the hydrophobic area can be away from the water. Okay, so this is what it is like. And you may also see that on the plasma membrane you'll find the channel proteins for transferring of the proteins. You'll be having the carrier proteins, then the glycoproteins, and the cholesterol, the peripheral proteins, and all. So these are the different proteins, the receptors, they act as channel passage pumps. There are various functions which these proteins perform, like diffusion, osmosis, and various other factors. And there's one more feature of the plasma membrane, and that is like they perform two major functions, that is endocytosis and exocytosis. Endocytosis is taking in the cell, like taking in the food material inside the cell, or engulfing the cell, the food material. Exocytosis, on the other hand, it is excreting or removing out of the cell. In an amoeba, for example, let's take an example of an amoeba. This is an amoeba, right? So in the case of an amoeba, a vacuole is formed when engulfing or capturing a prey. Example, paramecium. Like for example, if there is a paramecium near an amoeba, now this amoeba wants to digest or engulf this paramecium. What will it do? It will through its pseudopodia, it will move towards the paramecium. Then there will be a vacuole formed on its structure, and it will engulf the paramecium, and it will digest it. This is the functioning of the cell, because amoeba is itself a unicellular structure. So by this example you can understand that how endocytosis and exocytosis occurs. So this is what